Good morning. Today I'm going to discuss the topic transport of respiratory gases. The contents includes introduction, transport of oxygen in blood, oxygen dissociation curve, Bohr effect, transport of carbon dioxide, Haldane effect, chloride shift. Introduction. The transport of oxygen is the fundamental mechanism in aerobic respiration for the survival of the organisms. In the lungs, heart, vasculature, and red blood cells play an essential role in oxygen transport. In the lungs, oxygen diffuse out of the alveoli and enter into the capillaries which surrounds the alveoli. Then the oxygen is transported through the arterial blood that reaches to the interstitial fluid and then to the tissues. During resting time, our body requires 250 ml of oxygen per minute. Diffusion of gases. The diffusion or the exchange of gas occurs on the basis of diffusion, which again depends upon the concentration gradient. In the alveoli, the partial pressure of oxygen is 104 millimeter of mercury. And the blood which is coming from the systemic circulation, the partial pressure of oxygen will be 40 millimeter of mercury. So the exchange will take place by the process of diffusion, and the blood which is coming out of the venous end of the pulmonary capillary will have partial pressure 95 millimeter of mercury. And this is circulated through the general circulation, and the blood will be reaching into the tissues. They will be having the partial pressure of oxygen 95 millimeter of mercury, and the tissues will be having partial pressure of oxygen will be 40 millimeter of mercury. Again, due to the change in the concentration difference, the exchange of gas will be taking place, and the blood which is coming out of the venous end of the capillary will be having partial pressure will be 40 millimeter of mercury. Now we'll discuss how the transport of oxygen will be taking place in the blood. The oxygen is transported in two forms. One is in the dissolved form in the plasma, that is 3%, and the next one is it is bind with the hemoglobin, that is in the form of oxyhemoglobin for 97%. Dissolved oxygen in plasma. So 3% of oxygen is dissolved in the plasma. So that obeys the Henry's law. This law states that the amount of gas dissolved in a solution is directly proportional to its partial pressure. So large amount of oxygen can be carried in the dissolved form, provided the partial pressure of oxygen is sufficiently high. In combination with a hemoglobin, oxygen molecule combine loosely and reversibly with the heme portion of the hemoglobin. When the partial pressure of oxygen is high in the pulmonary capillary, oxygen bind with the hemoglobin. When the partial pressure is low in the tissues, oxygen is released from the hemoglobin. Oxygenation of hemoglobin. That means how oxygen is transported along with the hemoglobin. One molecule of oxygen can combine with one ion in the heme molecule. Since the hemoglobin contains 4 molecules of heme, so each molecule of hemoglobin can combine with 4 oxygen molecules. That combination is given below. That is Hb4 plus O2, that is forming Hb4O2. Again, to the Hb4O2, next oxygen is added. Okay. In this way, this reaction will take place, and that will be completed by Hb4O8. After the first step, that means when the first oxygen molecule combined with the hemoglobin, the affinity of the hemoglobin is increased to bind with the next oxygen molecule. This is because the binding of first oxygen molecule to the hemoglobin cause breakage of links in the hemoglobin and changes its configuration. So the configuration will change from the tensed state to relaxed state. That means there is TR interconversion. So there will be shifting affinity of the hemoglobin to bind with oxygen will be there. That means when the first molecule will bind with oxygen, followed by the second one, and then the third one. But the speed of binding will be increased one after the another. So because of this shifting affinity of the oxygen hemoglobin curve, it has given a sigmoid shape rather than a linear curve. Oxygen carrying capacity of hemoglobin is 1 gram of hemoglobin can bind with a 1.34 ml of oxygen. This is the diagram of oxygen dissociation curve. It has got an upper part that is called a loading zone and a lower part that is called a unloading zone. This curve is obtained when a relation between partial pressure of oxygen and percentage of hemoglobin saturation is plotted. This relation is not a linear one, but it is a sigmoid shape. And because of the sigmoid shape, it has got so many physiological advantages. That includes it allows greater uptake of oxygen at lungs in spite of greater variation in the alveolar partial pressure of oxygen. Tissues are supplied with oxygen according to the demand of the body. Hemoglobin can act as a buffer and maintain the tissue partial pressure of oxygen at about 40 millimeter of mercury. Oxygen dissociation curve has got two distinct zones. That is the top portion that is called the loading zone. It refers to the upper flat part of the curve and it is related to the oxygen uptake in the lungs. It is the part above where partial pressure of oxygen is 60 millimeter of mercury. An important fact regarding this loading zone is if the partial pressure of oxygen falls from 100 to 60 millimeter of mercury, hemoglobin saturation remains 90%. So this loading zone will provide high uptake of oxygen in the pulmonary blood, even when the alveolar partial pressure of oxygen is decreased in situations like when we are climbing mountains or when we are having some pulmonary diseases. So this is an advantage for the human being. Next zone is unloading zone. That is the lower part. The steep portion of the curve that occurs at the partial pressure of oxygen is below 60 millimeter of mercury. This part is concerned with oxygen delivery to the tissue and large amount of oxygen can be released from blood with a minor fall in the PO2. This property keeps the partial pressure of oxygen in the capillary blood relatively high and maintains a diffusion gradient for oxygen. At PO2 below 40 millimeter of mercury, 75% of hemoglobin is saturated. And the partial pressure of oxygen is lower than 40 millimeter of mercury. Larger amount of oxygen will be released and it is available to the tissues. Shift of oxygen dissociation curve. The oxygen dissociation curve can be shifted to right or left depending upon many factors. Several factors affect the affinity of hemoglobin for oxygen and shift the curve to either right or left. Shift to right. It indicates that decreased affinity of hemoglobin for oxygen, so large amount of oxygen is released into the tissues. The causes for the shift of oxygen hemoglobin curve to right side, that includes increase in partial pressure of carbon dioxide, increased hydrogen ion concentration, decreased pH, increase in body temperature, and increase in concentration of 2,3 dpg. Shift to left. It indicates the affinity of the hemoglobin to combine with the oxygen increases. So the oxygen will not be released to the tissues. The causes are just opposite of the other one. That is decrease in partial pressure of carbon dioxide, decrease in hydrogen ion concentration, increased pH, decrease in body temperature, decrease in concentration of 2,3 dpg, myoglobin, etc. Significance of P50. It means the partial pressure at which 50% of hemoglobin is saturated with oxygen. Normal value ranges from 25 to 27 millimeter of mercury. Hemoglobin affinity for oxygen is inversely related to the P50 value. That means Decreased P50 indicates increased affinity of hemoglobin and shift of oxygen hemoglobin curve to left side. Increased P50 indicates decreased affinity of hemoglobin and its shift to oxygen hemoglobin curve to right side. Bohr effect. Loading of carbon dioxide to the blood causes unloading of oxygen, a phenomena seen at tissue level. Thus decrease the oxygen affinity for hemoglobin. All factors which shift oxygen hemoglobin curve to right side decrease the affinity of hemoglobin for oxygen, and that all will produce the Bohr effect. The other gas which is transported is carbon dioxide. Again, carbon dioxide transport also will take place by the process of diffusion. The arterial end of the capillary will be having partial pressure will be 40 millimeter of mercury, and the tissues will be having 45 millimeter of mercury. So the exchange of gas will take place, and the blood which is coming out of the tissues will be having partial pressure will be 45 millimeter of mercury, and through the venous blood that will reaches to the alveoli, where the alveoli will be having partial pressure of carbon dioxide is 40 millimeter, and the blood which is coming will be having a partial pressure will be 45 millimeter of mercury, and the exchange will take place, and the blood which is going to the general circulation will be having the carbon dioxide partial pressure will be 40 millimeter of mercury. Transport of carbon dioxide in the blood. Carbon dioxide is transported in three forms. That is, first one is in the dissolved form, that is 7%, in the form of carbamino compounds, that is 23%, and in the form of bicarbonate, 70%. Dissolved form. The carbon dioxide is dissolved in the plasma as a solution, and that can be transported. In RBC, rapidly it will bind with carbon dioxide will bind with water and form carbonic acid, and then it is transported. Transport in the form of carbamino compounds. In plasma, the carbon dioxide combine with the plasma protein and form carbamino proteins, and it can be transported. In the RBC, the carbon dioxide combine with the amino group of the hemoglobin and forms carbamino hemoglobin, and then it is transported. Transport in the form of bicarbonate. Bicarbonate ions are formed in the RBC and they are diffused into the plasma. The filtered or from the blood, the carbon dioxide will be entering into RBC. Then it reacts with water to form carbonic acid in presence of carbonic anhydrase enzyme. Then it is dissociates into bicarbonate and hydrogen ions. The bicarbonate ions diffuse out of the RBC into the plasma, and then it is transported as sodium bicarbonate. Chloride shift. When the bicarbonate ions are moved out of the RBC, to neutralize the effect of this, chloride ions will diffuse from the plasma to RBC, and that is called the chloride shift or the Hamburger phenomenon. This is the carbon dioxide transport. From the blood, the carbon dioxide will be entering into the RBC. There it reacts with the water to form carbonic acid and dissociates into H plus and HCO3 minus. Thus, bicarbonate will be moving out, and that will be combined with the sodium and form sodium bicarbonate. Then, when the bicarbonate ions are moving out, to compensate the neutrality, again, the chloride ions will be moving back from the plasma to the RBC. Carbon dioxide dissociation curve. Thus, explain how the carbon dioxide is transported. It is obtained by plotting the relationship between partial pressure of carbon dioxide and total carbon dioxide content in the blood. In the body, the partial pressure of carbon dioxide value of the arterial and venous blood varies within a narrow range of 40 to 45 millimeter of mercury. The factors affecting carbon dioxide dissociation curve are one is the temperature and the concentration of the carbon dioxide. Haldane effect. Loading of oxygen to the hemoglobin in the lungs cause unloading of carbon dioxide. That means when large amount of oxygen is loaded, the carbon dioxide will be released from the hemoglobin. Thank you.